Hello and welcome to this eukaryotic cell biology video. I'm Dr. Aled Roberts, and in this video we're going to explore the structure of the nucleus, both in a general sense and some of the substructures which give rise to its important functions. Now, when we look at the eukaryotic cell, there are a number of differences which set it apart from its prokaryotic counterpart. However, one of the most influential differences is that eukaryotic cells contain a nucleus. In essence, it's the central compartment that safely houses the cell's genetic material and separates it from the chaos of the cytoplasm. This separation allows for a much greater level of control over gene expression and cell function, and so before we dive into its internal composition, let's first look at its general structure. And so starting with the size of the nucleus. Interestingly, the size isn't fixed. It can vary quite a bit depending on the type of cell. In most cases, the nucleus occupies around 10% of the total cell volume. Now, there is a general pattern here. The larger the cytoplasmic volume, the larger the nucleus tends to be, and the smaller the cytoplasmic volume, the smaller the nucleus will be. In other words, there's a sort of rough linear relationship between the volume of the nucleus and the volume of the cytoplasm. As one increases, so does the other. Now, in terms of actual numbers, the diameter of a typical nucleus usually falls between 2 and 10 micrometers, with an average size of around 6 micrometers. However, that's not the full story. The size of the nucleus can be influenced by other factors. Things like the number of chromosomes and the total DNA content of the cell can affect the size of the nucleus so that it doesn't conform to this proportional size. Now, what about the shape of the nucleus? You might be wondering, is the shape of the nucleus proportional to the shape of the cell? In other words, do round cells always have round nuclei, and do long cells have long nuclei? Well, not really. In most cases, the nucleus is roughly spherical, regardless of the overall cell shape. Take onion epidermal cells, for example. The cells themselves are rectangular, but the nuclei are still circular. Or if we consider liver hepatocytes, they can have quite an irregular cell shape, yet their nuclei are still large and round. So while there are some exceptions, the general rule is that the nucleus is kept to its spherical shape even when the cell doesn't. However, as is the case with most things in biology, there are always a few exceptions to these general rules. For example, if we look at this blood sample, we can see that not all nuclei are nicely round or neatly shaped. Take these neutrophils for instance. These are a type of white blood cell and their nuclei are both large, often occupying more than 10% of the cell, and instead of being circular, their nuclei are typically divided into multiple lobes that have thin connections between them. So whilst most nuclei tend to be spherical, some cells such as our neutrophils are highly specialised and serve as a great reminder that biological systems love to break the rules now and again. Finally, let's think about how many nuclei a cell has. Is the number of nuclei within a cell proportional? Or, to put it another way, does every cell just have one nuclei? Usually, yes. Most cells contain a single nucleus. And if we were to go back to our earlier examples, both of these typically have just one nucleus per cell. However, sometimes things aren't quite that simple. Under the microscope, you might see what looks like multiple nuclei. That can happen for a few different reasons. For instance, the cell might have recently divided and the two daughter nuclei haven't fully separated yet. Or what you're seeing might be a lobed nucleus, like what we saw in our previous neutrophils. Then there are cells that genuinely have more than one nucleus. For example, skeletal muscle cells are multinucleated because they're formed by the fusion of many individual precursor cells called myoblasts, and each one contributes its own nucleus. Some liver cells or hepatocytes can also be binucleate, meaning they have two nuclei. This helps them handle the high metabolic demands of the liver. And just to add another twist, not all cells have a nucleus at all. Mature red blood cells actually lose their nucleus during development. This gives them more space to pack in hemoglobin, making them highly efficient at carrying oxygen throughout the body. So whilst one nucleus per cell is the norm, some cells manage perfectly well with more than one and others function just fine without one altogether. Okay, now that we've looked at the size, shape and number of nuclei, let's explore the structure of the nucleus in a bit more depth. And we're going to move from the outside in, layer by layer, to see how each component contributes to the overall function of this essential organelle. So starting on the outside, we have the nuclear envelope. This is a double membrane that surrounds and protects the nucleus, separating its contents from the cytoplasm, creating a distinct environment inside the nucleus. This outer membrane is continuous with the endoplasmic reticulum and often has ribosomes attached, linking nuclear activity directly to protein synthesis and processing. Then embedded within the envelope are structures called nuclear pore complexes. You can think of these as highly selective gateways that control what goes into and out of the nucleus. Small molecules like ions and water can diffuse freely through these pores, but larger molecules such as proteins or RNA need specific transport signals to move across. Then inside the envelope we find the nucleoplasm, which is similar in consistency to the cytoplasm but tailored to support nuclear processes such as DNA replication and RNA transcription. Now, sitting within the nucleoplasm is one of the most prominent features, the nucleolus. This is the largest non-membrane bound structure inside the nucleus, and its main job to help manufacture ribosomes, the tiny machines responsible for protein synthesis. Finally, surrounding and interwoven throughout the nucleoplasm is chromatin, a complex of DNA and proteins which keeps the genome compact and organized, allowing it to fit inside the nucleus while still being accessible for transcription and replication. Now, when the cell is not dividing, chromatin appears relatively loose and spread out, allowing genes to be switched on and off as needed. However, when a cell prepares to divide, the genome is duplicated and the chromatin condenses further to form chromosomes. This condensation is crucial because it ensures that DNA can be accurately and evenly separated into two new daughter cells. Okay, so believe it or not, that was just an overview of the nucleus. We're now going to look at a few of the key structures in a bit more detail. So the nuclear envelope is as crucial to the nucleus as the cell envelope is to the cell, and that is because it defines the nucleus. It separates the nucleoplasm and everything within the nucleus from the cytoplasm and everything within the cell. And by doing so, it allows the nucleus to carry out its own highly optimized processes, things like DNA replication, transcription, and RNA processing, without interference from what's happening elsewhere within the cell. Okay, if we zoom in a little and take a closer look at the nuclear envelope, we can see that it has a double membrane structure made up of an outer and an inner membrane, which together form a clear boundary separating the cytoplasm from the nucleoplasm. Then between these two membranes lies the perinuclear space, a narrow gap of around 20 to 40 nanometers width. This space isn't just empty though, it's actually continuous with the endoplasmic reticulum, and so that close connection helps coordinate activities between the nucleus and the wider endomembrane system, linking processes like protein and lipid synthesis. Then on the flip side, the inner membrane is closely associated with a supportive network known as the nuclear lamina, which helps maintain the shape and structural stability of the nucleus. Now, scattered throughout the envelope are nuclear pore complexes. These structures bridge the inner and outer membranes and, more importantly, act as gateways regulating the movement of materials into and out of the nucleus. So the nuclear envelope isn't just a protective shell, it's a dynamic interface connecting the nucleus to the rest of the cell whilst maintaining a specialised and carefully controlled internal environment. Now, the nuclear lamina that I mentioned earlier is a mesh that lines the inner surface of the nuclear envelope. It's made up of intermediate filament proteins which form a sort of net-like structure just beneath the inner membrane, and this scaffold, as well as providing strength and shape, traps membrane-associated proteins, which in turn further stabilizes the nuclear envelope. The nuclear lamina also serves as a sort of anchoring platform, allowing various other elements to associate with that inner membrane, including specific structural proteins and regions of chromatin. And so these interactions help organize the contents of the nucleus and contributes to how DNA is spatially arranged. But besides trapping and anchoring components, what's the real purpose of the nuclear lamina? Why is it so important to the overall function of the nucleus? So the nuclear lamina actually has several important functions, all linked to where it's located and what it's made of. And so firstly, because it forms a dense mesh right beneath the inner nuclear membrane, it helps to maintain the shape of the nucleus and gives it mechanical strength. This support becomes especially important when the cell experiences physical stress or during movements of the nuclear envelope. Secondly, the lamina plays a key role in regulating DNA replication and in initiating the process of cell division. Certain proteins involved in these processes attach to the lamina, which helps coordinate when and where replication begins and ensures the nucleus can reorganize properly as the cell prepares to divide. The third function of the nuclear lamina is in organizing chromatin. It helps to anchor specific regions of DNA to the inner membrane, which in turn influences how chromosomes are arranged within the nucleus. This structural organization supports chromosome formation and helps regulate which genes are active or inactive in different regions of the nucleus. And finally, the fourth function, the lamina helps to anchor the nuclear pore complexes in place. This connection ensures that the nuclear envelope remains stable while still allowing efficient transport between the nucleus and the cytoplasm. So overall, the nuclear lamina isn't just a support net, it's an active and multifunctional structure that helps the nucleus maintain its shape, regulate replication, organize genetic material, and stabilize communication with the rest of the cell. So let's look at these nuclear pore complexes in a bit more detail. Now, it's actually one of the largest protein complexes in the entire cell, an incredibly intricate structure that's made up of around 500 individual protein subunits built from about 30 unique proteins, collectively known as nucleoporins. Now, these are organized into four main structural groups, each with a specific role in how the pore operates. So if we break this down, starting on the cytoplasmic side, we find the cytoplasmic ring. Now, as the name suggests, it's located on the cytosolic surface of the nuclear envelope, helping to anchor and stabilize the entire structure. On the nucleus side, there's a matching ring called the nuclear ring. It's built from the same types of nucleoporins as the cytoplasmic ring, but positioned on the inner surface of the envelope facing the nucleoplasm. Then between these two lies the pore ring, which forms the central channel of the complex. It's lined with specialized nucleoporins and contains a central transporter protein, which is what actively regulates the movement of materials into and out of the nucleus. And it's these that allow some molecules to pass freely while tightly controlling larger ones such as RNA and proteins. Now, extending inward from the nucleus side is the nuclear basket. This basket-shaped structure projects into the nucleoplasm and has several roles. It helps with gene tethering, anchoring the chromosome in place, gene silencing by deactivating certain genes, and interestingly, its structure can even influence the curvature of the nucleus itself, contributing to the common spherical shape of the nucleus. Finally, extending outward into the cytoplasm are the cytoplasmic filaments. These act as receptors for proteins and other molecules that need to enter the nucleus, recognizing them through specific signal sequences and guiding them towards that central transport channel. Okay, so the main function of the nuclear pore complex is to regulate movement across the nuclear envelope, both into and out of the nucleus. It controls transport from the cytoplasm into the nucleus and from the nucleus back into the cytoplasm. Now this movement must be tightly regulated. If molecules flowed freely, the nuclear barrier would lose all of its purpose. Okay, so inside the nucleus, the key process is transcription, making RNA from DNA. Many of the raw materials and enzymes needed for this, such as nucleotides and polymerases, are produced in the cytoplasm and must be imported into the nucleus through the pores. Meanwhile, translation, which is the building of proteins, takes place in the cytoplasm, and the mRNA blueprints for these are made in the nucleus, so they need to be exported out through the pores to reach the ribosomes in the cytoplasm. So in short, the nuclear pore complex acts as a highly selective gateway, ensuring that molecules move in the right direction at the right time and under strict control. Now typically, there are around 3,000 to 4,000 nuclear pore complexes per nucleus, but in more active cells where transcription and translation rates are higher, the number of pores will increase to meet that demand. And that is essentially the nuclear envelope and the nuclear pore. So for this final part of the video, we're going to turn our attention to the last key structure of the nucleus, the nucleolus. It's not only an essential part of the nucleus, but also the largest non-membrane bound organelle in a eukaryotic cell. However, before we can really understand what the nucleolus does, we need to take a short detour, a little journey into how DNA is condensed and organised within the nucleus. That background will make it much clearer how the nucleolus fits into the bigger picture of gene expression and ribosome production. And so within the nucleus we have DNA which naturally twists into a double-stranded helix. However, even though the helix is tightly coiled and condenses the DNA somewhat, the total amount of DNA in just a single cell is enormous. Now if we were to take all of the DNA within our cells and stretch it out end to end, it would measure between 1 and 2 meters in length, and it would need to fit inside a nucleus that's only a few micrometers in diameter. So clearly, the DNA can't just sit in there as one long thread. It needs to be condensed and organised in an efficient way. Now, to condense itself further so that it fits within the nucleus, the DNA wraps around special histone proteins. Now that might sound a bit counterintuitive, adding something to make DNA smaller. But the positive charge of the proteins counters the negative charge of the DNA, and this allows them to be compacted without repelling each other. This then allows DNA and histones to coil around one another, further condensing the DNA. As the DNA continues to stack and coil around each other, they form long fibres known as chromatin. Now this thread-like material is essentially a coil of DNA protein complexes, and each complex is itself a coil of DNA, and so by layering these coils, the cell dramatically reduces the overall size of its DNA, allowing meters of genetic material to fit neatly inside the tiny space of the nucleus. Now the chromatin is found throughout the nuclear plasm, distributed in regions that can vary in how tightly the DNA is packed, depending on the cell's activity. However, the highest concentration of chromatin is found within the nucleolus, which if you remember is a large membraneless organelle located inside the nucleus. Now the nucleolus contains two main components, fibrils and granules. If we look at this microscopic image, the fibrillar regions appear as strands of DNA that are actively being transcribed into ribosomal RNA. The granular regions, on the other hand, represent the next step in that process, where ribosomal RNA molecules are being packaged together with cytoplasmic proteins to form the building blocks of ribosomes. Now, just as some cells might contain multiple nuclei, some nuclei contain multiple nucleoli, with up to 4 per nucleus. Now the number tends to be consistent within a species, but can vary between different species, and it all depends on the amount of ribosomes the cells need to produce. Now, the nucleolus isn't a fixed structure. During cell division, the nucleolus actually disappears, and this happens as chromatin condenses into chromosomes and the cell temporarily pauses ribosome production. Then, once division is complete, the nucleolus reforms in each new daughter nucleus, ready to start the process all over again. And those are the main structures of the nucleus. From the nuclear membrane that creates a controlled environment, to the nuclear pores that control the flow of information, to the nucleolus that provides the machinery for protein synthesis, the nucleus is a system of remarkable precision and order. And with that, we come to the end of this video. I hope you have found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.